So what is the knowledge gap that your study is trying to do that no one else, or almost no one else, is trying to do? Or often you have the same question as 10 groups, but your method is different. So there are going to be some details or some perspective that only you, almost only you, will give to the field. What is that? And second, why is that useful? Now, I would say a generation ago, number one was enough. You were new. That was enough. I remember in my PhD, I worked on an enzyme that no one knew the structure. Maybe 3 groups were working. At that time, only one other group was working on it. And so I was able to do, even working on my own, I was able to do relatively simple studies and get published in good journals. Just because no one was working on it. It was really new. Was it useful? In a very basic research way, for some people, yeah. But because it was so different from other enzymes, good journals were interesting. For most of us, for most of you, those days are gone. Everything else you bring in, every additional protein, additional gene, additional method, has to be very tightly linked to that nucleus. Because as you know, if you remember from your high school physics, if I bring in details that are very distantly related to that, what happens to those electrons? They're more likely to leave. So the audience, when they get overloaded, they're going to do a memory dump. And what you want them to do is dump the details that are way out here, but not this stuff. So you look at gene X in your field. You're probably not the only lab looking at gene X. So when you get up and say, I'm looking at gene X, the audience is not necessarily going to be impressed or more interested because, we've heard about gene X. Another group looking at gene X. Your job is to actually very clearly demarcate what you are doing so that we know exactly where you fit in with the field. Because some of you are actually over here, right? I mean, hopefully most of you are over here. You have a part that's new, that sticks out of the field. It's not something that's already being done. For some of you, that's big. For others of you, I have an image here. What I'm trying to say is, right, some projects you're like this. Your new is maybe a little bit. Maybe some of you are really lucky or whatever. Your new is much bigger. We're not here to compare who's doing a better project. We're not here so that you present and you sound like you're going to cure cancer. What I do want you to be able to do in this workshop is, no matter how small this is or how big it is, the audience will see very quickly what this is. The second thing they've got to see immediately is, why is that useful? So you could say, okay, we're looking at gene X in the development of osteoclasts. Okay, we'll stick on bone for a little while. But you know that there are 10 other groups that work on that work on the same gene with the same cell type. So what are you doing that's different? Yeah, we're doing omics. And you think they should be impressed. Well, omics is not impressive anymore, right? Everyone does it, or you pay a company to do it. And then you want to say, all right, well, what we are doing that's different is we're looking at this gene in osteoclasts in early development or early differentiation of these cells. Because it turns out maybe in the field, most studies look in later development or differentiation. So what is your new here is in early differentiation. That concept has to come out over and over again. It has to be so obvious when the audience leaves the room, yeah, these guys are different because they're looking at early differentiation. Sometimes students say, you know, I understand what you're saying, but it's hard for me to simplify my project because my boss will get angry, or my project is very complex. I am not here to tell you to simplify your work, to dumb it down. Not at all. Because often, where your work needs to stand out is in the details. It's in the very technical. So that's why you have to be very good at doing this. And say, okay, the one thing that separates me from the others is the particular method. So that means that from an early point, I need to get to methods. I need to talk methodology, to talk technical. But I have to find a way to do it that's easy to digest, that the story can move forward. I'm not going to